Hey everyone, and welcome back to our channel. Today, we're diving into a fascinating technique that scientists use to untangle complex mixtures. Imagine you have a bowl of multicolored candies all mixed up, and you want to sort them by color. How would you do it? Well, in the world of chemistry and biology, scientists face a similar challenge, but on a microscopic scale. They use a powerful method called chromatography. So, what exactly is chromatography? Simply put, it's a technique used to separate different chemical compounds from a mixture. Think of it as a microscopic race. This method is incredibly useful, especially for separating things like proteins, which are the building blocks of our bodies. Amino acids, which are the components of proteins. And even the colorful photosynthetic pigments that plants use to capture sunlight. It's like having a superpower that lets you see all the individual ingredients that make up a substance. Now, there are several different types of chromatography, each with its own special twist. But today, we're going to focus on one of the simplest and most common methods: paper chromatography. It's a great way to understand the basic principles, and you can even try a version of it at home. Paper chromatography, as the name suggests, uses a special piece of paper. This process involves two key players: the stationary phase and the mobile phase. Let's break that down. The stationary phase is something that doesn't move. In paper chromatography, this is our piece of filter paper, which is made of cellulose. Think of it as the racetrack itself. The mobile phase, on the other hand, is what does the moving. It's usually a liquid solvent. This is the vehicle that carries our mixture along the racetrack. So, you have a stationary racetrack, the paper, and a moving vehicle, the solvent. So, how does the separation actually happen? It's all about a race. First, we take our sample mixture, let's say a drop of ink, and we place a small spot of it near the bottom of the filter paper. Then, we dip the very bottom edge of the paper into our mobile phase, the solvent, making sure the solvent level is below our ink spot. What happens next is pretty cool. The solvent starts to creep up the paper through a process called capillary action, just like how water travels up a paper towel. As the solvent moves up and passes over the ink spot, it dissolves the mixture and starts to carry it up the paper. Now, here's where the magic of separation begins. The different components within our ink mixture will travel up the paper at different speeds. Why? It all comes down to two things: how well each component dissolves in the solvent, and how strongly it sticks to the paper. Imagine our ink is made of a blue dye and a yellow dye. Maybe the blue dye is very soluble in our solvent and does unstick to the paper very much. It will happily ride along with the solvent and travel a long way up the paper. But what if the yellow dye is less soluble or sticks more tightly to the cellulose fibers of the paper? It's going to get slowed down. It won't travel as far. It's like a race where some runners are faster and some are slower. As the solvent continues its journey to the top of the paper, the different components of our mixture separate out into distinct spots at different heights. The most soluble and least sticky components travel the farthest, while the least soluble and stickiest components lag behind it. The end result is a series of colored dots, each representing a different chemical compound that was originally in our mixture. And this finished piece of paper, with all the separated components beautifully displayed as spots, has a special name. It's called a chromatogram. The chromatogram is like the finish line photo of our race. It gives us a visual map of all the different substances that were in our original sample. By analyzing the positions of these spots, scientists can identify the individual components of the mixture. It's a simple yet incredibly powerful tool that has revolutionized how we analyze substances in fields from medicine to food science and even forensic investigations. So, that's a quick look at the basics of paper chromatography. A stationary paper, a mobile solvent, and a race based on solubility and affinity. It's a beautiful demonstration of chemistry in action. I hope you found that interesting. If you enjoyed this video and want to learn more cool science concepts, make sure to hit that like button and subscribe to the channel. Thanks for watching, and we'll see you in the next one.